Okay, so hello everyone and welcome to BrainMap. It's my great pleasure to introduce our speaker today, Dr. Juan Helen Zhou. She's an associate professor at the Center for Sleep and Cognition and the deputy director at the Center for Translational MR Research at the Yung Lu Lin School of Medicine at the National University of Singapore. She's also affiliated with Duke NUS Medical School, and her laboratory studies selective brain network-based vulnerability in neuropsychiatric disorders using multimodal neuroimaging and machine learning approaches. She received her bachelor degree and PhD from the School of Computer Science and Engineering at Nanyang Technological University at Singapore, and she was an associate research scientist at the Department of Child and Adolescent Psychiatry at NYU. She did a postdoctoral fellowship at the Memory and Aging Center, University of California, San Francisco, and in the computational biology program at the Singapore MIT Alliance. Dr. Zhou is currently the council member and a previous program committee member of the Organization of Human Brain Mapping, and she serves as an editor of multiple journals, including Human Brain Mapping, Neuroimage, and Communications Biology. I would just like to remind the audience to please address any questions you have using the Q&A box. Dr. Zhou, thank you very much for coming here today or tonight, in your case. The virtual stage is all yours. Thank you very much.
Thank you so much, all. Yeah, very kind invitation and introduction. And good morning, everyone. My name is Helen Zhou, and it's my great pleasure to be here. And thank you all for joining us during this tough period. In the next 45 to 50 minutes, I'd like to share with you our work on human brain network breakdown in early neurodegenerative and cerebrovascular disorders from a longitudinal perspective. And you feel free to interrupt me if you have any questions. Given the aging society worldwide, the number of people with dementia will roughly double every 20 years. Alzheimer's disease, or in short, AD, is the most common type of dementia. In 2010 and 2013, Jack and his colleagues proposed a hypothetical model of the major AD biomarkers as shown in this figure. The y-axis represents the biomarker abnormalities and x-axis represents the clinical disease stage. It starts with extracellular amyloid beta plaques, then intracellular neurofibrillary tau tangles, followed by brain atrophy, even during cognitive normal stage. These pathophysiological abnormalities precede memory decline and clinical symptoms by many years to decades. Given this model and the failure of multiple drug trials targeting late-stage dementia, the long-term goal is to predict disease vulnerability and progression from the clinical and neuroimaging examination years before the appearance of any symptoms at the individual level. To achieve this goal, today, as a first step, I will first introduce network-based neurodegeneration framework using network-sensitive imaging methods. Then, under this framework, I will focus on three key questions highlighted here. You can see that, are you able to see the first line here? Or is blocked by the...
Yes, we can see the Alzheimer's disease slide.
Okay, so we're going to ask three key questions. First, are we able to detect symptom-specific changes in neurodegenerative and cerebrovascular disease subtypes? And second, can we track the longitudinal changes at pre-dementia stage and the influence of risk factors? And lastly, can we review disease mechanism and predict disease progression? To start, here are the structure T1 weighted structure images of patients with 5 different neurodegenerative syndromes. As you can see, all 5 disorders have very distinct atrophy patterns. AD on the top usually lost their episodic memory and visual spatial functions, cause atrophy in a posterior parietal, posterior cingulate, and hippocampal temporal network. In contrast, behavior variant frontotemporal dementia, or in short, BVFTD, disrupt the social emotional functions and target an anterior frontal insular, anterior cingulate cortex, and subcortical network. Based on the evidence from neuropathology, imaging, and animal models, neurodegenerative disorders have been thought to relate to network-based dysfunction. In other words, each disorder targets a specific large-scale neural network. In 2008, Bill Seeley, my mentor at UCSF, led us to systematically test this network-based neurodegeneration hypothesis in living humans for the first time. We used a network-sensitive neuroimaging methods, which include resting state fMRI and structure MRI. Resting state fMRI, or task-free fMRI, captures the blood oxygenation level dependent signals fluctuations at rest condition. The intrinsic connectivity networks derived from such data can map temporal synchronized, spatially distributed, and spontaneous low-frequency both signal fluctuations. Besides the low-level auditory somatomotor networks, we can also derive the higher-level cognitive networks, such as default, control, salience, or attention networks. Starting with the atrophy pattern highlighting the salient color of each disorder, we pick the most atrophied region, as circled here, and performing seed-based functional connectivity network mapping in healthy participants. The resulting intrinsic functional connectivity networks is highlighted in yellow color here. You can see great similarity between functional connectivity networks in healthy controls in yellow and the atrophy patterns in disease in cyan color. Similarly, if we use the same seed and perform structural covariance mapping, basically look at how gray matter volume co-vary across participants in a group of healthy controls, we found very similar patterns highlighted in green here. Our findings support that there is a direct link between the functional networks and the gray matter structure in health. And we also confirm that regions that wire together within each network, they fire together, they're connected together, and they grow and shrink together. So taken together, we confirm network-based neurodegeneration hypothesis in living humans. This work leads to many interesting questions on selective network vulnerability along disease progression. Following Jack's model, yours and other colleagues proposed and refined the hypothetical models of AD. They include the resting state fMRI as well as FDG PET metabolism. So, can we detect the symptom-specific changes in dementia subtypes? The first question. Alzheimer's disease targets the deformer network specifically. This is supported by increased amyloid burden from PET, atrophy from structural MRI, and hypometabolism in the deformer network using FDG PET. In 2004, the pioneering work done by Microvascular from Stanford found that Alzheimer's disease patients have reduced deformer network connectivity derived from resting state fMRI, which is independent of brain atrophy. So how about the other dementia subtype? Indeed, our previous study published in Brain have demonstrated that AD and BV-FTD featured the divergent functional connectivity changes in the default and salience network. These are the two most common disorders for ages less than 65 years old. BV-FTD have a reduction in the salience network, but increased the functional connectivity in the deformer network. In contrast, Alzheimer's disease, as you could expect it, they have reduced deformer network connectivity. But in contrast, they have higher functional connectivity in the salience network. This divergent pattern corresponds very well with their symptom deficit profile of AD and FTD, and again supports the network-based dysfunction hypothesis. Since then, this finding has been replicated in quite a number of studies, including amnestic MCI, APOE4 carriers. And one step further, my team hypothesized that the network coordination between these two networks, as well as the subcortical regions, could be differentially disrupted in AD and FTD. So we are not looking at intra-network FC, we are looking at between-network coordination. So this is based on a Singapore data set. We test this hypothesis by systematically study the whole brain functional connectivity in AD and FTD. Based on the whole brain postulation, what we found is that AD and FTD have this disrupted community structure compared to age-matched healthy control at the whole brain level. But if we narrow it down to each individual networks, as you can see here and highlighted in the square box here, AD have very specific fragmented modular structure in the default and control network, while FTD have more severe damage to the modular structure in the salience network and subcortical regions. Importantly, such connectivity profile track the disease severity and clinical symptoms. In our previous brain paper, across BV-FTD patients, lower salience network functional connectivity in right frontal insula, y-axis, was associated with more severe disease severity measured by clinical dementia rating sum of boxes on the x-axis. And similarly, when we consider inter-network connectivity, we found that increased salience network participation coefficient, indicating a loss of modular structure and functional distinctiveness, was related to more severe behavioral problems measured by neuropsychiatric inventory score across AD and FTD patients. These findings support the distinct loss of network integrity in AD and FTD, underlying their opposite cognitive and behavioral symptom profile. When we focus on Alzheimer's disease alone, recently, several works have tried to explain the clinical variability in AD. However, these studies only examine AD patients without comorbid conditions. But in reality, we know that AD frequently coexists with cerebrovascular disorders. In short, CVD. AD with CVD is the most prevalent dementia subtype, especially in Asia. On its own, CVD is associated with decline in executive function and processing speed. And CVD biomarkers or MR markers include lacunes, white matter hyperintensity, microbleeds, etc. CVD and AD are suggested to have additive effects on cognitive decline. So for the same clinical severity of dementia, AD-only patients present with more severe AD pathology like amyloid and tau than those comorbid with CVD. Therefore, Dr. Joanna Chong from my lab asked this question: would we see a similar or differential functional network breakdown patterns in AD patients with and without cerebrovascular disease? Based on the memory clinical cohort from Singapore, for the first time in the field, we showed that AD patients with and without cerebrovascular disease featured a divergent functional connectivity phenotype. We focus on the deformal network and executive control networks based on their symptom profiles. When we compare AD with and without CVD with the same disease severity head-to-head, AD had more reduction in the default mode network connectivity, Lower connectivity in the deformed network is also associated with lower hippocampal volume, which is a marker for AD pathology. Similarly, the CVD groups have higher frontal executive control network connectivity, which was correlated with greater burden of white matter hyperintensity measured here in the y-axis. So this work supports the differential network degeneration phenotype in AD with and without CVD, which reflect their underlying pathology. Besides functional network breakdown, white matter degeneration is also an early event in AD, which closely follows the A-beta deposition. Diffusion MRI, as you all know probably, measures the movement of water molecules within the white matter tracts, and it can be used to examine white matter microstructure and structure network connectivity. However, the traditional single tensor diffusion model suffers from partial volume effect. In other words, in some voxels, the signal there is a mixture of tissue compartment and water content. So how can we resolve this problem? So Dr. Alpha Postolak from your center has proposed a biotensor free water method. This method separates the signal of each voxel into two compartments. The tissue compartment represents exon bundles. The free water compartment represents extracellular water. So reduced FET represents the axon damage and demyelination, while the increased free water represents the vascular damage or neuroinflammation, possibly due to blood-brain barrier leakage. So Jifang, from my lab, we have the opportunity to collaborate with Alpha Postolak, and we are interested in the differential white matter changes in AD with and without cerebrovascular disease. Compared to the original single tensor results, as you can see on the top row here, the differences between ADs with CVD compared to AD are highlighted in the red color. The free water corrected tissue compartment revealed more accurate and focal tissue damage, which is located in the frontal and occipital lobe in AD with CVD compared to AD without CVD. When we look at the free water increases, it is not surprising that the AD with CVD and vascular dementia have widespread free water increases in white matter, representing vascular damage. But interestingly, when we look at AD without CVD, they also have higher free water widespread compared to age-matched healthy controls, even when they actually have comparable CVD burden measured by white matter hyperintensity compared to control. And note that we actually derive the free water from normal appearing white matter. So we exclude those regions with white matter lesions. So such free water increases could be due to inflammation or microvascular changes. Moreover, when we derive a mean free water increases in normal appearing white matter, they were associated with symptom severity, CDR sum of boxes, across all patients, while the original white matter hyperintensity ratio does not. Building upon our previous study, Jifang continued to investigate whether such brain free water increases are related to cardiovascular markers and whether it could mediate the relationship between cardiac function and future cognitive decline. So this is a fresh data from the lab. Based on a memory clinical cohort from Singapore, what we found is that the higher cardiovascular markers, such as high sensitivity cardiotropin P, was indeed related to free water increases in widespread white matter and network-specific gray matter regions, including default, control, attention, and sensory motor networks, highlighted in red here. More importantly, the association of the higher cardiovascular marker with the longitudinal decline in executive function and verbal memory up to 5 years were mediated by the higher white matter free water in a global manner and network-specific, the free water increases in gray matter, specifically the default mode network for the memory and the executive control network for the executive function. So these results underscore the important role of free water related to vascular modulation. So in summary 1, I hope this finding, based on the structure, diffusion, and functional MRI, have already lent support to the network-based dysfunction hypothesis at dementia stage. And brain network phenotype is able to explain the clinical variability, track symptom severity, and relate to AD and vascular burden. Till now, I have reported most of the cross-sectional work in the dementia stage. As you know, to facilitate early detection and intervention, it's important for us to move to the left-hand side of this curve, the preclinical and pre-dementia stages. So we are interested in the longitudinal brain network changes in these 2 stages, as well as the influence of risk factors and pathology. Building upon our previous cross-sectional work, looking at divergent functional connectivity in AD with and without CVD, we collaborate with Dr. Zhou from South Korea to investigate this question further. Because previously, we actually did not quantify the AD and vascular pathology. We simply divide them into 2 groups. Now, we have the opportunity to objectively measure the amyloid burden using PET in vivo and vascular pathology using MR markers. And we are interested in the longitudinal changes in the default and executive control networks over time in patients with mild cognitive impairment, which is considered a prodrome stage of dementia. Zhou and Chong test the hypothesis that amyloid and CVD exert divergent influence on the functional connectivity changes over these 2 networks over time. We compare the 2 pure group, basically pure subtypes, which is amnestic MCI A beta positive and subcortical vascular MCI A beta negative. What we found is that the prodrome patients with pure amyloid burden showed a steeper decline in the deformal network functional connectivity over time, as you can see in the lines highlighted in red. When we look at the prodrome patients with pure cerebrovascular burden, which is the subcortical vascular MCI A beta negative, they show this steeper increase in the executive control functional connectivity over time. Again, this replicate our previous work and demonstrate the differential network degeneration phenotype in the prodrome stage that is related to amyloid and the CVD separately. You may ask, okay, how about those mixed groups? We have amnestic MCI A beta negative. We also have subcortical vascular MCI A beta positive. While we include those 2 groups with mixed pathology and repeat the analysis in a continuous manner from low to high PIP SUVR, from low to high LACUMs, we also found the same thing. So this finding actually highlight the divergent influence of amyloid and CVD pathology on brain functional network trajectory over time in pre-dementia stage. So now, let me move to the very early preclinical stage. The key question we ask here is that, are we able to detect the network changes early, and whether the longitudinal trajectory can reflect the cognitive decline over time, and what is the potential influence of risk factors such as ApoE4 genotype. Now I'm going to take 1 minute to talk about modular organization of the brain. We know that human brain is functionally organized in a highly modular and distinct manner, with high connectivity within networks representing functional specialization and low connectivity between the networks representing functional segregation. This modular structure can aid specialized and integrated information processing and increase the robustness and adaptability of our brain networks. For example, the task-positive networks and task-negative networks are anti-correlated or segregated. So task-negative network refers to deformal network. The task-positive refers to salience and executive control and attention networks. As you can see, the salience network is closely connected with executive control and subcortical regions, while these two networks are often anti-correlated with the deformal network. Given the multidimensional nature of symptoms in neurological disorders, we hypothesize that both within and between network connectivity will be important for cognition. To cover this gap, Eric Ng, postdoc in my lab, studied the longitudinal changes in functional connectivity and cognition over 4 years in a group of Singaporean healthy older adults aged 50 to 85. We found that there is a functional connectivity decline within the default and executive control network over time. And each line represents one participant, and the higher value here represents better functional specialization. So you can see everybody is getting, having a loss of functional specialization. When we look at the relationship between the default and the executive control network, we see very interesting age-dependent loss of functional segregation between the two networks. And then Dr. Zhou Yingwei, together with Eric, we studied the influence of ApoE4 genotype. So ApoE4 carriers, when we divide them into carriers and non-carriers, we see that the ApoE4 carriers who were at higher risk of developing dementia, as highlighted in the red line here, had faster loss of functional segregation over time compared to the non-carriers in green color. This demonstrates that ApoE4 carrier basically have this accelerated aging pattern. When we look at the relationship of these functional connectivity profiles with behavior, interestingly, basically we found that the greater loss of functional segregation between the two networks over time was associated with faster decline in processing speed on the y-axis over time. And very recently, we also replicate this data from a whole brain level because we would like to know whether it is only because of default and executive control network. How about salience? How about the rest of the sensory motor networks? So we basically include everybody, include the cortical and subcortical regions, and we look at how the modular structure change with aging. So again, this is the work by Joanna Chong. What we found is that elderly show this cross-sectional and longitudinal decline in the network segregation and distinctiveness. So compared to young, they have a lower local efficiency and higher participation coefficient. And such pattern also keep changing over time. And if we do look at all 9 networks of covering the whole brain and do multiple comparison correction, what we can find is that such aging trend is specialized or localized in the higher level cognitive networks, including salience, executive control, and deformal network. Let me change gear again to talk about early white matter degradation in pre-dementia stage. Again, we are interested in the influence of CVD and amyloid burden. Ash white pin from my lab again use free water imaging in collaboration with Alpha Pastelak. We use the data from ADNI. What we found is that among a group of cognitive normal elderly, if we divide them into amyloid positive and amyloid negative, the amyloid positive cognitive normal elderly showed a greater reduction in fractional anisotropy in the right uncinate fasciculus over time than those who do not have amyloid. This is suggesting that there is an amyloid-related early white matter degeneration in preclinical stage. In terms of CVD burden, in collaboration with Netherlands and Christoph Chen, we are interested in this very novel cerebrovascular marker, so-called cerebral microinfarcts, or in short, CMI. CMI is more prevalent in Alzheimer's disease and related to cognitive deficits. Animal models have suggested that the cortical CMIs induce white matter impairments, neuronal loss, and decreased neuronal response in remote cortical regions, even beyond the original cortical CMI region. So based on this finding, we hypothesize that CMI may affect the cognition via disrupting local and long-range white matter pathways. Li Wenzhang from my lab actually tested this hypothesis in both preclinical, prodromal, and dementia stage. What we found is that the presence of cortical CMIs lead to lower structural network efficiency and degree centrality in the executive control ECN and dorsal attention network. This is especially true in the pre-dementia stage, and this effect is gone in the dementia stage. And such structural network breakdown also mediates association between CMI and cognition, which support the hypothesis that CMI influence the cognition via remote regions, white matter tract breakdown, and the influence take place earlier. Lastly, we hope to build up the disease progression predictive model based on multimodal brain measures. So one question is whether such brain network measures can give us insight into the disease mechanism and progression. We have demonstrated this possibility at the group level previously by comparing healthy connectivity graph measures to disease severity, atrophy severity in disease. Basically, in Alzheimer's disease, what we found is that regions having shorter functional paths to disease epicenter have higher atrophy in disease. If we extend this across all 5 dementia subtypes, we actually validate this finding from the shortest path to epicenter to in-house and total flow in-house. So this finding fit best with the transneural spread model and wear and tear model. It means that key network hubs or epicenters exist within each disease-specific network, and the closer you are to the disease epicenter, the more traffic through the region, the more vulnerable you are going to be in the disease. So this finding is supported by previous work which demonstrate that the degree centrality is correlated with amyloid deposition, which support nodal stress model. Another study also support the Prion-like transneural spread model, which basically says that the closer you are to the epicenter derived by diffusion tensor imaging. So note that this is structural connectivity this time. It is associated with earlier amyloid arrival basically. Recently, Jesse Brown from the UCSF extended this framework to the individual level. So all previous studies are based on group level. This is the first study which is able to show that rather than the group level disease epicenter, we can find a unique atrophy disease epicenter at the individual level and use this particular patient-tailored disease epicenter. We're able to predict the future longitudinal atrophy based on the individual connectivity patterns. So this finding again support the transneural degeneration hypothesis. Under this framework, my team has also made some effort to derive the individual level network integrity score and evaluate whether and how the influence of the network-based neurodegeneration on cognitive function vary with different cognitive stage. Given the hypothetical AD models, the impact is definitely nonlinear. So to answer this question, Qianxin in my lab, they derived the individual level metabolic network score from FDG-PET and the structural covariance network score from structure MRI. As you can see, they resemble the key canonical brain networks. Using partially square and time-varying coefficient model, we demonstrate this nonlinear stage-dependent association of hippocampal and default network breakdown with memory impairment in AD continuum. And such network breakdown framework is also useful when we try to digest the disease progression in stroke patients. Based on the longitudinal structure MRI data from 3 months to 1 year post-stroke, we found that faster degradation of brain structure networks was associated with greater longitudinal decline in attention, language, and memory, suggesting the potential of using brain network integrity to predict and monitor disease progression. So I'm going to skip this. Basically, this is another work shows that the baseline functional connectivity and structural connectivity can predict the psychotic conversion in persons at risk for psychosis, based on a study from Singapore. Note that this observation of unifocal connection-based spread is only one of the possible network breakdown scenarios. It does not take into account the genetic vulnerability or other biological factors. So it is the staggered multifocal onset, which is not related to connectivity, is very possible. Or it could be a combination of the two. So we need to integrate neuroimaging, genetic, and pathological data from both animal and human models to further test this hypothesis. So in conclusion, I hope I have convinced you that it is important to examine early and longitudinal brain network breakdown trajectories in neurodegenerative and cerebrovascular disease. And moving forward, we definitely need a multimodal neuroimaging methods together with other assays from blood, CSF, or genetics. We also need machine learning and computational approaches to increase the reliability and discover robust and valid features with higher accuracy, generalizability, and interpretability. So one example is we need to look at dynamic functional connectivity because we know that the static functional connectivity derived from resting state fMRI can be confounded by many stuff. And there is this time-varying recurring connective patterns that is related to the varying level of consciousness, vigilance, and individual behaviors. And you're welcome to check out our previous paper on this. With that, I'd like to thank the funding agency and all the collaborators and my lab members who have contributed to these studies. And thank you for your attention and happy to take any questions.
Thank you very much, Professor Zhou, for this great talk. And we will now move to the Q&A phase. So anyone, if you have any questions, please use the Q&A box. On the meanwhile, I'll start just by being curious to your next research plans or just the next steps, or what we should expect seeing from your lab soon.
Well, I think the next step will be to continue to So actually, I'm very excited about upcoming. Basically, we recently have been funded to do a large-scale clinical trial to basically targeting vascular dementia. And as I have mentioned, we are very interested in this type of disease. And we are going to do the clinical trial, try to slow down the progression of this disorder from persons at risk for dementia. And we are going to collect multi-modal neuroimaging, genetic and blood data, and try to see whether we can predict who is going to benefit from this intervention and why. And obviously, machine learning is going to be used. And another effort we are trying to do is definitely, you mentioned machine learning in the introduction, and I haven't talked about it because we would like to actually develop machine learning to actually help us to, let's say, segment some of the cerebrovascular biomarkers from the image, which is still very troublesome these days. And we hope to collaborate with other collaborators. So if you're interested in any of the projects, feel free to contact me. If you have any question, obviously you can also email me. Yeah.
Great. Thanks. So we have a first question from Dr. Olivia. Just a second, I'll try and see if I connect her. But on the meanwhile, she's saying just a very interesting talk. What are your thoughts on how the variability in anatomy in AD confounds the bold signal and might in part cause variability in the RSF MRI data? For example, how does atrophy and partial volume affect the fMRI signal?
So you're asking about the, so the question is in the Q&A, or I cannot see the question?
Yes, I'm just gonna try and allow Dr. Olivia to talk, maybe just a sec. Yeah, it's in the Q&A anyway.
I can't see the questions.
Just a sec. So Dr. Olivia, you can unmute, I think, and then we can see you, hopefully. On the meanwhile, I'm just going to repeat it. She says that, what are your thoughts on how the variability in anatomy in AD confounds the bold signal and might
okay, I think I can speak now, right?
Sorry.
Okay,
yeah, great.
Yeah,
a very interesting talk. I very much enjoyed this. I have a question about the results you were showing earlier, where you showed how basically networks that were wiring together are also the ones that you find to grow and shrink together.
Yeah.
Do you have any thoughts on how atrophy and partial voluming in your fMRI acquisition might also induce some variability in your resting state data? And do you have some knowledge what really underlines the atrophy or the anatomical changes in terms of, is it a vascular change or a neuronal change?
Very good question. So when we look at the functional connectivity changes in this dementia populations, we always have to control for gray matter changes at the voxel level. So all my results presented here have been controlled at the voxel level or at least the network level to basically indicate that the functional connectivity changes are partially independent of the atrophy. Because if you don't have neurons there, obviously your functional connectivity is not going to be very high. So we would like to rule out that possibility.
How do you do it in practice? How do you control for it, if I may ask?
We do a CAT-12 to derive the gray matter probability maps. And then we can actually use, previously we used a toolbox called SPN N or something, or biological mapping in SPM that actually allow us to voxel level control. But nowadays, I think FSL randomized also allow us to do that, basically entering the gray matter map at the individual level as additional nuisance variable at the voxel level.
Okay, so it's like a statistical, you take it as a confounding factor into your statistical analysis?
Yes.
Okay, thank you.
Great. Next question from Iman. Iman, can you unmute?
Oh, I can see the question now. Yeah.
Yes.
Did you see?
Yeah.
Please, yeah.
Yeah, thank you very much for your very interesting talk. I was wondering if you saw any increase anywhere that was associated with a disease that could possibly mean a compensatory effect or a resiliency effect in the patients, Alzheimer's disease patients.
Yes, yes. So the two study I mentioned just now, I think they could be compensatory. One is in the early AD patients. In the very first study, in the brain study 2010, we found increased connectivity in the salience network in the Alzheimer's disease patients and increased connectivity of the deformer network in the BVFD patients. So note that these two patients, this group of patients, when we recruit them, they are still at early stage, so they don't have a lot of atrophy yet. So we think that it is kind of compensatory given the reciprocal relationship between the salience and the deformer network. So that's one example. And the second example, remember our CVD studies, we found this increased frontal executive control network in the AD patients with cerebrovascular disease. And what we think is that it could be compensatory, because what we found is that this increased functional connectivity in the frontal executive control network is associated with greater CVD burden, right? So because, you know, so but obviously this is a kind of speculation, right? Whether it is indeed compensatory, we definitely have to check. You know, in terms of the other mechanism.
Okay, thank you very much.
Yeah, and
Thanks.
sometimes you can see the U shape, right? The increase and later on decrease. Yeah.
Great. Next question from a video, please. Video, can you unmute?
Yes, yeah, very interesting talk. I was interested to ask you about cerebral vascular disease and small vessel disease and cerebral microinfarct. How do you segment and quantify this and the burden of these diseases? And if you use any clinical classification rating scales to, you know, for these biomarkers, how do you integrate and use this information together with the other biomarkers? Are you using AI? Are you using more classical machine learning or manual or a combination of everything?
So the findings I reported here are still based on the visual rating of the CVD. As you know, CVD actually includes 1 meter happy intensity, which could nowadays, there is some automatic segmentation toolbox, but they are still not very accurate. And so there's a visual rating of 1 meter happy intensity, and then basically there are certain criteria, say age match the 1 meter happy intensity rating greater than 8, then is considered CVD. And then we also count number of lacunes, number of microbleeds, etc. And so this is based on some published standardized criteria. So you're welcome to check our paper. So all of these are based on visual rating, but there are ongoing challenge, ongoing effort, you know, to basically try to segment or quantify these automatically. But I would say, you know, a lot of work still required. Yeah, so basically, let's say, you know, if we want to quantify, you know, how the number of lacunes related to the functional connectivity, then we can only, you know, use number of lacunes. We're not able to use the location of the lacunes, right? However, you may suspect the location of the lesions could also play a role in terms of how the network is disrupted. In fact, there's some newer study showing that in stroke patients, if you look at their infarcts and see how infarcts is located with reference to 1 meter networks, and to evaluate their impact score, it is going to be related to their, let's say, the recovery. So I do think we need a bit more AI and other approaches to try to stratify the patients more.
Okay, thank you. So at the moment you say you rely also on human experts, for all the studies that you have done, and you are still doing. You don't discard the human expert from your analysis, and you are still relying on them when you.
Yeah. Yes. Yes.
Okay.
Because you need the ground truth to develop machine learning approach as well.
Okay.
Thank you. The next question from Martin Zhang. Martin, can you unmute?
Yes, I'm muted. Hi, Dr. Zhou. A very nice talk. So it seems you are from the Center for Sleep and Cognition. For Alzheimer's disease, have you tried any studies on sleep in connectivity abnormality of Alzheimer's disease in different stages of Alzheimer's disease?
So you're asking whether I have measured sleep at different stage of Alzheimer's disease, or looking at how sleep relate to connectivity?
Correct.
The latter one or the first one? Maybe I can answer both.
Maybe it's general.
Yes. Although I come from Center for Sleep and Cognition, I'm not a sleep expert. So I don't do much work on sleep. With that being said, my colleague, Professor Michael Chi, is really a world-leading expert in sleep. And currently, I think he is collaborating with my close colleague, Christoph Chen, to look at the role of sleep in Alzheimer's disease and the relationship with amyloid and imaging. If you are asking about whether we have checked the functional connectivity, patterns at sleep deprived people, yes, we have checked. Someone believed that sleep deprivation could be an aging model, right? So we have checked so that PNAS paper, and I mentioned at last, is actually performed in the sleep deprived participants. And we asked them to perform resting state as well as auditory vigilance task. And we found also a loss of functional segregation in these participants. And they have this a big scene of different dynamic connected states. When their brain is highly vigilant, maybe I can go. Okay, maybe I shouldn't. I have a very nice video shows that when they are highly vigilant, their brain is nicely segregated and specialized, right? So the default and the task positive networks are anti-correlated. When they are very sleepy and less vigilant, and such pattern disappeared. There's no anti-correlation in their brain.
Thank you.
Okay, so I don't see any other question yet. So I would like to thank you so much, Dr. Zhou, for staying up so late for us. And we hope to see you in person soon. And again, thanks for this great talk.
Thank you all. Thank you everyone for coming. And I very much enjoyed it. And welcome to Singapore. When all the dust is settled. Okay. Yeah, take care. Stay safe.
Thank you.
Okay.
Thank you very
Bye.
much.
Bye.